Okay, guys, this is our first lecture video of our online class. I hope that the message that I posted on Google Classroom uh, about what we're going to be doing in the class made sense to you. If you haven't filled out that survey that I posted this morning yet, please do that for me because that's going to let me know if you're going to need any copies of the materials that we're going to be using for class. It's also going to let me know if you would rather continue to do the videos and or rather maybe try Google Meet. If you have an iPhone, you can get Google Meet on your iPhone. Um, you can probably also get it on your laptop or your home computer, and we can play around that if you would rather do that. You'll just have to let me know. We're going to go with whatever the most people decide because I really can't do both because that would take an enormous amount of time for me to make a video for each day and then turn around and sit for an hour and a half on doing the same lecture, on a, live on Google Meet. So, we're going to go with whatever the majority of you decide. Because I thought maybe you might like to do the live in-person lecture better. That way you could ask questions, which normally you don't in class, but I thought you might want to. But then I also considered that maybe the video would be more helpful to you in that you wouldn't have to be available at this certain time every day. You could do it on your own time and answer the questions as well as go back and rewatch the video if you wanted to. Maybe even more than once if you wanted to stop it and take notes or rewind it and go back and listen to something again. Either way, as I said in my message, either way you choose to do it, I will still be available for questions. You can ask questions here on Google Classroom. You can email me with questions that you have. You can, email me and I can call you, or we can Skype or FaceTime or something like that if you have questions, because I know a lot of this material is difficult, especially in this chapter. But even more than that, it's just a lot of material. So I am here if you need help. Um, Also, I hope you understood the other things that I was saying about um, the lecture quizzes and exams, the questions that I'm going to ask you, because there's no real way for me to police what you're using when you take the test, whether you're using notes or uh, looking things up online. So you can expect that the questions that I'll be asking you for quizzes and tests are going to require you uh, to think, to be able to think about the information and apply it to different situations. Probably not going to be at least some of the questions will be questions where you won't just be able to look up an answer online. You'll have to actually be able to think about the material and come up with an answer. Um, Also, in lab, again, we're not going to be able to do lab practical as long as we're out of school, so we won't be getting to look at models of the muscle and the spinal cord and things like that at this point. We're scheduled to be out for 2 weeks. I don't know if that will end up being longer. I know some schools are out for 3 or 4 weeks. Uh, I've heard recommendations of doing this isolation for 8 weeks. So at this point, it's for 2 weeks, and that may be all it is. And so in 2 weeks, we would be back to school and everything would be like it was. But we just don't know at this point. So at this point, I had thought about maybe putting lab off until we got to come back to school. But then again, like I said, I don't really know for sure when we'll get to come back to school. Again, it's for 2 weeks right now, and I hope that's all it is. But, time will tell. But I had thought if we would definitely be back in 2 weeks, we could just maybe go through some lecture, and then we could do all the labs when we, make up all the labs when we come back. But yet again, like I said, if it ends up being longer than 2 weeks, we may end up having to, not having any time that we would be back to make up all the labs that we would need to make up. So I thought we'll just go ahead and stick with the syllabus the way it is, and we'll do labs on Thursday and Friday. You will need to still go ahead and print out those labs and complete the diagrams and the questions, so that if we were to get to come back in 2 weeks, you would have those already completed so that you could use those on a lab quiz. Uh, However, if we don't get to come back, that will still be good practice for you. That will be something you'll be able to use, uh, when we have online quizzes. And again, as I said, when we do have a lab exam, if we're not back to school by the time we have our lab exam, again, there's really no way for me to police what you're using, at least not that I know of at this time, what you're using to do your exams, both your lecture exams and your lab exams. So the diagrams that I give you, if we have to do a lab exam online, the diagrams that I give you, you will have diagrams that you'll have to complete or that you'll have to answer questions about. Those diagrams will not be the exact same ones that you have on the lab pages that I post on Google Classroom for you, or on the lab handouts that you have. It won't be the exact same diagrams. The lab exam will cover the same information, but it won't be the exact same diagrams. Now, quizzes, it probably will be, because you're allowed to use your lab handouts on those anyway. But for exams, the questions that you have, or I'm sorry, the diagrams that I use will not be the exact same diagrams. It'll be the same information, the same material, but it won't be the exact same diagrams. The diagrams will look different. So anyway, you can let me know if you have any questions about anything. At this point, we're all just kind of flying blind, because we've not had to do this before, and we really don't know how long this is going to last. So we're just going to make the best of it. And as I said, if you have any questions, concerns, please let me know. I don't want to make this any harder on you than it already is, but yet I do have to make sure that you are not just, looking at all this on a web page or in your notes and not really understanding or learning the information. So I will have to, put some questions and diagrams on quizzes and tests that are going to test more than just your ability to look at your notes or the internet to get the information, but that will actually test your understanding of the material, and that you are listening to the lectures, studying the material, and understanding what it is that you're learning. Okay, so let's go ahead and get started with, I had on your syllabus that we were only going to spend 3 days on Chapter 9, but there's just really no good way for me to be able to do that. So we're going to have 4 days on Chapter 9, which will put us, we'll finish Chapter 9 on Tuesday, then on, we'll actually finish Chapter 9 on, we'll finish Chapter 9 on Tuesday, I'm sorry. We'll do a little bit with Chapter 10 on Wednesday, and then review. I'll go over some things that you definitely, need to be able to answer questions about on the test on Wednesday as well. And then on Thursday, we will be having a lecture exam. At this point, of course, it will be online, probably a Google form. And you will need to not only know the information, but to be able to apply that information that you are supposed to know, so that you can demonstrate your understanding to me. And then on Friday, if you'll remember when we had our lab exam, I gave you guys lab handout 12 to go ahead and start on. So you should already have that in your binder. And then we will also you'll be finishing up the lab handout 12 and also be completing lab handout 13 on Friday. So again, if you are going to need copies of that, I will put it on Google Classroom. If you won't be able to print it out on your own, like at home, then you can just fill out that survey and let me know that you will need copies of that. And I don't exactly know how yet that we're going to be getting materials that students need to their homes, but I will talk to people, and I will post on Google Classroom or some way to let you know exactly how we're going to make sure that you can get the materials that you need if you're going to need materials from me. So please, as soon as you can, maybe after you watch this video lecture, or as soon as you can, complete that survey for me, so that I know and I can make sure I get those materials and get them to the whatever place that I need to get them to, so that you can get them, whether it is you picking them up in the front office or whether it is we're delivering them to you. Again, I don't exactly know how that's going to work yet, but if you'll fill out that survey as soon as you can and let me know how that's going to work, or if you are going to need copies, then I will let you know as soon as I can, how I will get those copies of these materials to you. And again, as I said, if you have any questions, please call the school. please email me. Please send me a message on Google Classroom, and I will answer it as quickly as I can and the best that I can. And again, don't worry. It's going to be a little trial and error here at first, but we will get all this figured out and make it work. All right, so last week we began talking about muscle contraction. We learned about how the action potential is transmitted from the nerve cell to the muscle cell, and then how that muscle contraction results in, how that nerve impulse, that action potential, results in a muscle contracting. And we learned about on Wednesday the excitation contraction coupling. Again, talking about both the mechanical and the electrical components of muscle contraction. And so now we're going to start stepping back a little bit more and looking at, first of all, here, how can we affect the amount of tension in a muscle, and then also looking at different types of muscle fibers that we have in our bodies. And then we'll go on tomorrow and we'll look at how how the contraction of skeletal muscle is different from that of smooth muscle. So we'll look a little bit more about smooth muscle, and I think cardiac muscle as well tomorrow. So we're going to start out today by looking at a twitch. We learned about this in anatomy last year. Remember, a twitch is it's one contraction, it's one phase of contraction relaxation. It is one action potential that causes a twitch. There are three phases of a twitch. You have your lag phase, your contraction phase, where the tension rises, and then the relaxation phase, where it falls back to the resting tension in the muscle. Again, this little twitch is something that mainly exists in a lab, in muscle. All the useful movements that we make do not involve a single twitch. Again, a single twitch is a single cycle where the stimulus is applied, and then you have your contraction and your relaxation. We can look at what happens during each one of these phases during this lag or latent phase. This begins when the action potential is transmitted from the axon, from the nerve cell to the muscle cell across that synaptic cleft. Then that action potential moves across the sarcolemma down the T-tubules to the sarcoplasmic reticulum. So all of that would be included under the lag phase. During contraction, this is when the calcium is coming out of the SR, and the calcium binds to the troponin pulls the tropomyosin away, and the actin binds to the myosin, and you get the cross bridging and the sliding of the actin past the myosin filaments. So all of that is happening during contraction. And then during relaxation, this is when the ATP attaches to the myosin heads. They release from the actin. The calcium is pumped back into the sarcoplasmic reticulum, and the actin filaments slide back out to their resting positions. And of course, the tension falls in the muscle fiber. We can look at two types of muscle contractions. Those are isometric and isotonic. With an isometric contraction, the muscle does not change in length and the resistance does not move. So there is more tension in the muscle. The muscle is contracting, but it really doesn't change in length, and whatever you're trying to lift doesn't move. So for example, if you're trying to lift something that's too heavy for you, that is an isometric contraction. Like if you were trying to pick up a car. Normally you can't pick up a car, and so you are pulling against this resistance, but your muscles don't shorten and your resistance does not move. Even though you're generating tension in the muscle is not changing in length overall, and the resistance, which would be the car that you're trying to lift, is not moving. An isotonic contraction, on the other hand, the muscle does shorten and the resistance does move. So when you are lifting something that you can actually lift, that you can move, like lifting your pencil or lifting a book, then you're doing an isotonic contraction. You are increasing the tension in your muscle, the muscle is shortening, and the resistance, which would be the book that you're lifting, is actually moving. Now, there are two different types of isotonic contractions. Those would be concentric and eccentric contractions. Again, the concentric contraction is one where you are overcoming the resistance and the muscle is shortening and the object is moving. So that would be lifting a book. An eccentric contraction, the tension is maintained, not increasing, not increased, but maintained, and the muscle actually lengthens, like when you're lowering the book or when you're lowering a heavy object, like lifting weights and then lowering the weight. When you lift the weight, you're doing a concentric contraction. The resistance is moving, your muscle is shortening, and the tension is increasing. When you are lowering the weight back down, that is an eccentric contraction. The tension is being maintained, but the muscle, instead of shortening, is actually lengthening. Okay, it's also important for us to look at a motor unit. He talked about this on the video that we watched last week. A motor unit is a single motor neuron and all of the muscle fibers that it controls. So, if we look at like a classroom, we have a teacher that's teaching and we have students in the classroom. So that could be considered a motor unit. The teacher is the neuron, it's the nerve cell, and then the students are all of the muscle fibers that are being controlled and regulated by that nerve cell. So when we take this neuron and an action potential moves down that neuron, all of the muscle fibers that are being stimulated by that neuron will all be stimulated at the same time. So this is a motor unit, this is the neuron, and you can see how it's branching out and innervating all of these different muscle fibers in this muscle. And each one of these, notice that these are fascicles, and there's several muscle fibers in the fascicle, and you have several different axon terminals here, several different terminals that are innervating. So each terminal would innervate a different muscle fiber. So when we have an action potential that moves down this neuron, it goes to all of these branches and all of these axon terminals, and it stimulates and causes contraction of all of the muscle fibers. So we call it a motor unit because a single action potential in this nerve cell stimulates and causes the contraction of all these muscle fibers that this one nerve cell innervates. So again, one action potential in this neuron causes the contraction of all the muscle fibers in this motor unit. They all are going to contract at the same time as a result of an action potential that is moving down this neuron and being transmitted to these muscle fibers. And we can actually look at the size of motor units. Large motor units are motor units where you have a single neuron that is innervating hundreds of muscle fibers, whereas a small motor unit is going to be one where a single neuron is innervating only a few muscle fibers. So in general, we tend to have large motor units in large muscles, and it takes very strong stimuli to cause these large motor units to contract all of the fibers in that unit. You're going to have less precise control over these muscles that contain these large motor units. So, for example, the muscles in your thigh, such as your sartorius and your gracilis and your quads, they are going to also in your abdomen, your obliques, your abs. Those muscles are large muscles. They have large motor units, so a single action potential is going to stimulate hundreds of muscle fibers to contract, and you're not going to have really precise control. You're going to have a lot of tension, but not a lot of precise control over those muscles. However, small motor units are found in small muscles, and so a single action potential down that neuron is going to stimulate only a few muscle fibers to contract, and you're going to have a lot more precise control over those small muscles with those small motor units. And it doesn't take a lot of a very strong stimulus to cause that those muscle fibers in that motor unit to contract, and so you're going to have more precise control over those muscles. These are going to be muscles like in your fingers, the muscles that control your eye, and we'll talk more about those muscles that control your eye later, but those are small muscles with very small motor units. So weak stimuli will cause those to contract, and so you're going to have more precise control over those. We say that muscle fibers respond to a stimulus in a graded fashion. Basically, the amount of force or tension generated is going to depend upon the number of cross bridges that are formed. The more cross bridges that we can form between actin and myosin, the more force or tension that we're going to generate in that uh, with that contraction or during that contraction. So there are three things that can affect the number of cross bridges and therefore the amount of tension that we can get during a contraction, and that is going to be how frequently we stimulate the muscle fiber. The more frequently or the more often we stimulate the muscle fiber, the more tension that we're going to be able to generate, because we're going to be able to form more cross bridges between actin and myosin. The muscle fiber diameter, the larger the diameter of the muscle fiber, the more myofibrils, which means more myosin and actin, so more cross bridges. And then the fiber length at the time of contraction, we'll see that how far is that muscle fiber stretched before we try to contract it. We'll see that there is a maximum length, or an optimal length. If it's too short, it won't contract as well. If it's too long, it won't contract as well. There's an optimal length of that muscle fiber, and therefore the sarcomeres in that muscle fiber that will let us get a good contraction. So we're going to first look at letter A here, the frequency of stimulation. And there are 4 things that we can look at: TREP, or it may be called TREPI, I'm not sure about that. Wave summation, incomplete tetanus, and complete tetanus. And we did talk about these 3 in regular anatomy. Now, we didn't really mention this one too much, but we did talk about the other 3. So looking at TREP, basically this is, basically what you see here is TREP, where, or TREPI, I guess you could call it, where we do one stimulus, we have that one twitch, contract, relax, and we allow it to relax completely. Then immediately after relaxation is over, there's another stimulus. Notice what happens. The tension goes up a little bit higher. If we do that again, we let it relax completely. Then we do another stimulus. Then the tension goes up even higher again. So it's kind of like a staircase. That's why we call it a staircase process. The muscle fiber, the big thing here that makes it different from the other things that we're going to look at, the wave summation, the incomplete, and the complete tetanus, is that the muscle fiber is allowed to relax completely. So the muscle fiber relaxes completely, and then immediately another stimulus is applied. This causes that tension to rise just a little bit. Again, we're getting more cross bridges here. Basically, what's happening here, you allow the muscle to relax completely, but if you do another stimulus immediately, there is going to be an overall increase in calcium that has been released out of the sarcoplasmic reticulum, because if you deliver that stimulus immediately after relaxation, then there hasn't been time for the sarcoplasmic reticulum to reabsorb all that calcium. So, we relax it, the sarcoplasmic reticulum starts reabsorbing calcium, but it doesn't have time to reabsorb all of it. So when we stimulate it again, this causes more calcium to be released, and so the amount of calcium overall goes up. And so we have more cross bridges forming, which means more tension. And then if we let it relax completely, the sarcoplasmic reticulum starts absorbing calcium again, but again, it doesn't have enough time to reabsorb all that calcium before we stimulate it again. And so when we stimulate it, more calcium is released, which causes an even bigger increase in the amount of calcium in the muscle fiber. That released out of the sarcoplasmic reticulum. And then, of course, that calcium ends up allowing the myosin to bind to the actin. And so we get even more cross bridges and more muscle tension. So, again, the difference between this one and the others is that the muscle fiber is allowed to relax completely before we apply another stimulus. However, because we do have an increase in the calcium being released from the SR, because it wasn't able to reabsorb it all before we stimulated it again, then we end up with more cross bridges being formed and an increase in tension, as you can see here. The next one is wave summation. Basically, we're going to just increase the frequency, or how often we're stimulating the muscle. So with wave summation, we're not going to allow the muscle fiber to relax completely. We're going to allow it to contract, but then before it is allowed to relax completely, then we're going to stimulate it again, which is going to cause an even bigger increase in the muscle tension. Again, more cross bridges are going to be formed because we're not even allowing it to relax completely. So we're not even going to allow that SR any time to absorb calcium before it releases more. And so we're going to be increasing the amount of calcium in the cell, in the intracellular area, and that's going to allow more cross bridges to be formed. Incomplete tetanus, the main difference here between incomplete tetanus and wave summation is that we are going to reach a maximum tension. We do still have some relaxation going on here, but it reaches a maximum tension. So basically at this point, when you're in incomplete tetanus, you have reached that maximum tension. You have opened all of the active sites on actin, and you have the maximum number of cross bridges that can be forming. Okay, so that's how you reach this maximum tension. So once you've reached maximum tension, you have the maximum number of cross bridges that can be forming. Okay, you can't get any more than that. The only difference here is that we are still allowing some relaxation to occur. Okay, finally, with complete tetanus, remember we have reached maximum tension and there is no relaxation. So we are stimulating this muscle cell so frequently that it doesn't come down at all. There's no relaxation. It goes up and it stays up. So the stimulus is applied, and it's being applied so often that there's no going down at all of this graph. No relaxation happens at all. And so this gives us the maximum tension with no relaxation at all. That is complete tetanus. So again, with TREP, we are allowing it to relax completely in between stimuli. With wave summation, and again, that increases the tension, more cross bridges are forming. With wave summation, we are still allowing it to relax, but not to relax completely, which increases the tension even more. With incomplete tetanus, we are still allowing it to relax some. It's not completely relaxing, but it is relaxing some, but we do reach a maximum tension here. And then with complete tetanus, what makes it different from incomplete is that, okay, we're still reaching that maximum tension, but we have no relaxation at all. So that is the highest tension that we can get when we're not allowing it to relax at all, and we are reaching this maximum amount of tension. Okay, again, as far as diameter, I said this earlier, the bigger the diameter of the muscle fiber, the more force that can be generated because a bigger muscle fiber has more myofibrils, which means more actin and myosin. Because the myofibril is bigger, it has more actin and myosin in it, because that's what the myofibril is made of. And so with more actin and myosin, you can form more cross bridges, which means more tension during contraction. Okay, and then the last thing that we said that could affect the amount of tension that we get in the muscle is the length of the muscle at the time of contraction. We have what we call active tension, which is the force applied to an object when a muscle contracts. And this active tension can increase or decrease depending upon the length of the muscle fiber in the muscle. As the length of the muscle fiber increases, the active tension also increases up to a point. So again, if the muscle fiber is not stretched at all, you're not going to get a lot of contraction because the thick filaments are touching the Z discs, which they're not supposed to be doing that. Okay, you only have actin filaments attached to the Z disc, and the myosin filaments, the thick filaments, are kind of inside of those two Z lines or Z discs. But if you have it not stretched at all, it's like all crushed together, then it's hard for those myosin filaments to attach to the actin because they're touching those Z discs at the end of the sarcomere. But if we stretch it, we start stretching the muscle fiber out, then that starts, it gets those thick filaments away from the Z discs, but we still have lots of zone of overlap. We still have lots of overlap between actin and myosin. So that's our optimum length. When we have optimal overlap between actin and myosin, we can get the optimum number of cross bridges forming. But then if we keep stretching it out beyond that, then we start decreasing that zone of overlap. And the more we decrease that zone of overlap, as we stretch that muscle out and decrease that zone of overlap, then that means fewer cross bridges can form because actin and myosin aren't overlapping as much. And again, that leaves us with very little contraction that can happen. We also have passive tension, which is the tension applied when the muscle stretches but is not stimulated, and this exists because muscle and its connective tissue do have some elasticity. So when the muscle is not contracting, it's actually stretching and not being stimulated. You do still have a little bit of tension just because you have all that connective tissue and stuff in the muscle that's kind of keeping the muscle from stretching too far out. And so that's called passive tension being applied to the object. And then the total tension is the sum of the passive and active tension. So active tension is the force being applied when your muscle contracts. Passive tension, the muscle is not contracting, it's not being stimulated, it's just tension that's being applied to the object, just like where you're holding it. You're not really contracting, it's just that the connective tissue in the muscle just has a certain length, and it keeps the muscle from stretching out too far. So here is a picture that really kind of shows you this. Here is our optimum length. We have lots of zone of overlap here, so we're going to get lots of cross bridge formation, the myosin heads attaching to the actin. Here we have too much overlap, and we have these thick filaments that are kind of coming, almost touching those Z discs, which kind of interferes with the attachment of the heads to the actin. Here we don't have any zone of overlap. The muscle is so far stretched that there's not any zone of overlap, and we can't really do much contraction at all. So again, too much overlap to the point that thick filaments are touching the Z discs, we don't get as much contraction. Too little overlap, and we really get very little contraction at all. And then we have this optimal amount of overlap. So an optimum length of that muscle and that muscle fiber gives us the greatest tension in the muscle. Okay, so the force of contraction in a muscle as a whole increases as the action potential frequency, or the frequency of stimulation, how often we stimulate it, increases. But again, that doesn't really count for very much of the tension in the muscle, in the whole muscle. What accounts for most of the tension in a whole muscle depends on the number of muscle fibers that are being stimulated. Okay, that are again goes back to those motor units. The larger the motor unit, the more muscle fibers that are being stimulated, the more muscle fibers that are contracting, the more tension that we can get. The smaller the motor unit, the fewer the number of muscle fibers that we're contracting, and so the less tension we're going to get. So the number of fibers that we are stimulating to contract is going to account for most of the tension that you're generating in your whole muscle. The how often it's being stimulated accounts for some of the tension, but most of it is accounted for by the number of fibers that are being stimulated. And again, remember that it's the nervous system, the neuron, that is determining how many muscle fibers are getting stimulated, whether you are stimulating those small motor units in a muscle or the larger motor units in the muscle. So the strength of the contraction then is graded. It can range from weak to strong depending upon the strength of the stimulus. So we have what we call multiple motor unit recruitment. The strength of the contraction depends upon the recruitment of motor units. Remember, a single muscle is going to have many motor units. Again, a smaller muscle will probably have more smaller motor units, whereas a larger muscle will have probably a lot more larger motor units. But again, we're going to see that with recruitment, we're going to stimulate the smaller motor units in that muscle first, and then we will bring in the larger motor units as we need more tension generated in the muscle. However, again, we have to go back and think about the stimulus. We have to reach threshold in order to get that muscle contracting. So we actually have 4 different results as during multiple motor unit recruitment. Again, it's graded from no response up to maximum response. So no response is going to be a subthreshold stimulus. A subthreshold stimulus is a stimulus that is below. It is subthreshold. It's below threshold. And so it is not strong enough to cause an action potential, and so we're not going to get any contraction. So here we go. A subthreshold stimulus, a stimulus below threshold, we get no contraction. A threshold stimulus is here. We get a twitch, okay? It is strong enough to produce an action potential and cause the contraction of all the muscle fibers in that motor unit. So this is a threshold stimulus. We are getting contraction of all the muscle fibers in a motor unit. Then we have submaximal, which means below maximum. So basically, if we get a stronger stimulus than a threshold stimulus, something stronger, then we're going to get more tension in the muscle. Basically, the reason we're doing that is because we are bringing in additional motor units. So again, if we need more tension in the muscle, then we're going to, this submaximal stimulus tells that muscle, hey, we need more tension. And so that submaximal stimulus is going to recruit additional motor units. It's going to stimulate additional motor units in that whole muscle so that we can get more tension being produced by that muscle. And then we have what we call a maximal stimulus. This is where we have a stimulus that's strong enough to produce action potentials and stimulate all the motor units in the muscle, and that gives us a maximum amount of tension in that muscle. So when we are stimulating, when we have a stimulus that is maximal, it is strong enough to stimulate every single motor unit and every muscle fiber in every motor unit in that muscle, then we get maximum tension in that muscle. We can't get any more tension than that maximum tension. And we get that maximum tension as a result of a maximal stimulus, a stimulus that is strong enough to stimulate, to generate action potentials in every motor unit, in every muscle fiber, in every motor unit in that whole muscle. Now, again, remember, all these action potentials are the same. What we're talking about here is the stimulus. The stimulus is, the touch of the spider on your knee versus the touch of the elephant on your knee, like you talked about in the video last week, okay? The touch of the spider on your knee, maybe that is a threshold stimulus, or maybe a submaximal stimulus. It's enough to generate action potentials in enough motor units that you can flick that spider off of you, okay? But a maximal might be when you have the elephant on your knee, okay? It is a stimulus. That elephant, that pressure on your knee is a stimulus that is strong enough to generate action potentials in all the motor units in that muscle. Like, let's say a thigh muscle or something. It's strong enough to generate action potentials in all the motor units in that muscle. So you're going to have every muscle fiber and every motor unit contracting, and that's going to generate a maximum amount of tension in that thigh muscle, let's say, maybe in one of the quads or something. So again, we're not talking about varying the strength of the action potential because all action potentials have the same strength. What we're talking about is varying the strength of the stimulus, varying the strength of, let's say, the pressure that's being put on your knee or something like that. So varying the strength of the stimulus can result in different amounts of tension, all the way from no tension in the muscle to maximum tension in the muscle. Again, different stimuli can generate different amounts of tension depending upon how many motor units in that muscle are being stimulated. And again, this is called the size principle. Smaller motor units are recruited first, followed by larger motor units. So if we have the spider on our knee, then we're probably going to stimulate small motor units. That's probably, like I said, it's going to be maybe a threshold or a submaximal stimulus. And we're going to stimulate small motor units that are going to get that spider off of our knee. But if we have that elephant on our knee, then, okay, we're going to have to bring in larger motor units. So we're going to stimulate the smaller ones first. That's not going to be enough. Then we're going to stimulate larger ones, and it's that maximal stimulus, that stimulus of that big old heavy elephant's foot being on our knee, that is going to generate action potentials in all of the motor units in that thigh muscle and give us enough tension, hopefully, to move that elephant foot off of our knee. Not a whole elephant, just an elephant foot. Okay, we also talked about muscle tone last year in regular anatomy. Remember, this is the resting tension in a muscle. Even when you are at rest, you do have some motor units in your muscles contracting. So it's the constant tension in a muscle. And again, it is over long periods of time. So even when you're at rest, some of your motor units in your muscles are contracting. And they are contracting out of phase with one another. Okay, so you'll have a few that are contracting at one point, and then those will rest, and then some more will start contracting. So they're not all contracting at the same time because that would generate a lot of tension. They're just going to have a few generating off and on while you're resting, keeping tension in that muscle. This helps to keep your limbs in the right position, to keep you from hurting yourself when you're sitting or standing or laying, whatever. It helps to hold your head upright, keep your abdomen flat. It helps just to hold your body parts, your muscles and bones and things in a good position to help avoid damage. And remember, when you pass out, all muscle tone is lost. Okay, looking at the two different types of muscle fibers that we have. The first one is called the slow twitch or oxidative fibers. I think we called these slow fibers last year in regular anatomy. Remember that they respond more slowly to a stimulus, and so they contract more slowly. They are going to be smaller, and they also have a better blood supply. So they are going to use aerobic respiration, which means with oxygen. If you have blood, then you have oxygen, so you can do aerobic respiration. And again, that happens in mitochondria. So these fibers have a better blood supply. They have more mitochondria, so they are doing more aerobic respiration. And because they're doing aerobic respiration, they're not producing lactic acid, so they don't fatigue as quickly. So they are fatigue resistant. And again, they are red. If you remember, they're red because they have a better blood supply, and they also have large amounts of this pigment called myoglobin. It's very similar to hemoglobin, and it helps them to hold on to oxygen and to be able to do that aerobic respiration. So it's the blood and the myoglobin that makes them appear darker red. You have certain muscles in your body that have more of these red, slow fibers. Your muscles that help you maintain your posture. In your lower limbs, you're going to have probably a lot of these. And then the dark meat of the chicken leg and thigh are going to be slow fibers. And then the duck breast, okay? A chicken breast is white. We'll talk about white in a minute. But the duck breast is dark. It's red fibers because ducks don't really fly a whole lot. So therefore, they don't need, they fly more. They fly more. And so they do a lot more aerobic respiration to be able to fly for longer periods of time. And so they have a dark meat breast. And then if you are a long distance runner, then you would have more of these dark slow fibers or red slow fibers in your lower limbs than someone who is a sprinter, let's say, because long distance runners need to do that aerobic respiration. They need to be fatigue resistant because they are running more slowly for longer periods of time, like running a marathon. You need to be able to run more slowly for longer periods of time. So these type of muscle fibers allow you to do less powerful contractions for longer periods of time. So again, a duck flies for longer periods of time, a dark breast. Long distance runners run for longer periods of time. So that's the dark, slow red fibers. The fast twitch or type 2 fibers, we also talked about these. Last year we called these the fast fibers. They are going to respond more rapidly to stimuli, and they are going to contract more quickly. The myosins that are in these fast twitch fibers can break down ATP more rapidly. And so they are going to have large deposits of glycogen, which is energy storage, an energy storage molecule. So because they need that energy very quickly, because these fibers are going to be doing those brief, powerful contractions. They don't have as much blood supply, and so they're not going to be red. They're going to be white. So this is going to be your chicken breast, because again, chickens don't fly for long periods of time. They only do brief, powerful movements to get away from a predator. So this is why the chicken breast is white, because it has a lot of these fast twitch fibers. They don't have a lot of mitochondria. The ones that they do have are smaller because they're not doing aerobic respiration. They're doing anaerobic respiration. They don't have a lot of oxygen associated with them because they have less of a blood supply. So they don't have a lot of mitochondria because they're not doing aerobic respiration as much. They're doing more anaerobic respiration. And so they can contract, again, more powerfully, but for shorter periods of time. The reason for shorter periods of time is because they're doing anaerobic respiration, they're producing lactic acid, and so they fatigue very quickly. So you can use these fibers, again, for brief, powerful contractions, and then they're going to be fatigued. They're going to be worn out. They're going to have a buildup of lactic acid and start cramping. Again, someone who is a sprinter, who is running very hard, very fast, okay, that is going to be, they're going to have more of these fast twitch fibers in their lower limbs. Most people have fast twitch fibers in their upper limbs. Most everybody does, because you don't use your arms for, extended periods of time. You may use them for brief, powerful contractions, and then you're not using them as much. There are two forms of these fast twitch fibers. I think last year we called one form, we called the 2B the actual fast fibers, and we called the 2A the intermediate fibers. But the 2A are fast twitch oxidative glycolytic fibers that are FOG. Fast twitch oxidative glycolytic. These use both anaerobic and aerobic respiration. So again, they're intermediate. They use anaerobic so they can contract more powerfully, but they also use a little bit of aerobic as well, so they can last a little bit longer. They don't fatigue as quickly. The fast twitch glycolytic or FG fibers are exclusively anaerobic. They just use anaerobic respiration, and so therefore they are going to be the ones that are going to fatigue the most quickly. They can produce very powerful contractions, but for very, very short periods of time. Okay, and you can actually train so that you can enlarge one or the other. Most people have both types, but again, it's a lot of this is genetic, so it can vary. Some people may have more of one type, some people may have more of another type, but you can actually train your muscles to cause one or the other to enlarge. So, for example, weightlifters, they enlarge their fast twitch because they're doing those brief, powerful contractions to lift these very heavy weights. Or people who run sprints actually train their muscles to have more, to enlarge these fast twitch fibers. Whereas when you do aerobic exercise, you actually enlarge slow twitch fibers. Again, because you are doing those less powerful, more sustained movements using that aerobic respiration that causes your slow twitch fibers to become enlarged. So training can enlarge one or the other. We know that exercise affects the size of our muscles just in general anyway. If you exercise a lot, if you use your muscles a lot and you stimulate them very frequently, then you are going to experience what's called hypertrophy, which is an increase in the size of the muscle and the muscle fibers. You basically are going to cause those muscle cells to form more myofibrils. You're going to have more satellite cells that will fuse with the muscle cells to make the muscle itself larger. You'll have more nuclei, which means more production of protein, which means more actin and myosin and more myofibrils. And you're also going to have an in strength. On the other hand, atrophy is, we talked about this last year as well, as a decrease in muscle size. Remember, if you don't use it, you lose it. So when you don't use your muscles on a regular basis, they are not stimulated as often. The muscles actually decrease in size. You're going to have a decrease in myofibrils. And this is reversible, as we said last year, up to a point. When you get to the point in severe situations where the muscle cells actually die, then this atrophy becomes irreversible. Last thing we want to talk about in this video is that exercise also affects, when we exercise, we have a lot more muscle contraction. And when muscles contract, they generate heat. And so this not only increases our body temperature, this is why we get hot when we exercise, because our muscles are contracting so much more and all that contraction is releasing more heat. But it also increases our metabolic rate. And so this is why exercise helps us to burn more calories, burn more fat, because we're increasing our metabolic rate. Even after exercise, our metabolic rate stays high due to oxygen debt, due to the fact that, our cells are trying to catch up with all the oxygen that we are requiring to be used to do that aerobic respiration. And then after a little while post exercise, you actually you're going to end up increasing the amount of heat that you're losing, because your body is trying to cool down because you generated all this heat from muscle contraction. You're so hot, so remember, your blood vessels in your skin will start dilating to release this extra heat. Also, you'll start sweating, and that evaporation of that sweat will cool you down, and both of those things helps you to release a lot of the heat that you've built up. So for a while post-exercise, your metabolic rate will remain high, but then it will go back down as the excess heat is being lost due to vasodilation and sweating. And on the other end of the spectrum, Our muscles can also help to bring our body temperature up if we get too cold. Not only will we have the blood vessels in our skin constricting, remember that's called vasoconstriction, and that's kind of what our skin does for us, but our muscles actually, if we get too cold, we will begin shivering. Well, what is shivering? Shivering is just kind of the uncoordinated, uncontrolled contraction of muscle fibers. Uh, and when those muscle fibers are contracting uncoordinated, uncontrollably, that causes that shivering and shaking that you experience. But the purpose of it is to generate heat because when your muscle fibers, again, when they contract, they produce heat. So if you're too cold, if you get really cold, then you will experience this uncoordinated contraction of muscle fibers trying to generate heat. If you get too hot, then whenever you're exercising, your muscles contracting, you get really hot because when muscles contract, they produce heat. And again, you have to get rid of that extra heat through vasodilation and sweating. So your skin is really helping you get rid of that extra heat. But if you don't have enough heat, not only can your skin try to conserve heat, but your muscles can actually generate heat when they contract. Okay, and that is all that we're going to talk about for today. Again, you can go back and listen to the video again if you need to. If you have any questions about anything, please don't hesitate to email me, and I can try to answer you in an email, or if you need me to call you, then you can just send me your number in an email, and I can call you. We can FaceTime or whatever if you have any questions. Also, don't forget to answer, you should have already answered the entrance slip questions before you started watching this video. Those questions are about last Wednesday's lecture. And then don't forget today to answer your exit slip questions and submit those to me as well. Also, don't forget, again, one more time, remind you to do that survey for me so that I can get an idea of whether we want to continue doing the videos or whether you would rather try to do, some live lecture through Google Meet. If you have any questions, again, please just email me and let me know, or email me with your number and I can call you. I hope you have a good rest of your day, and I'll be talking to you soon. Bye-bye.